Welcome to the poster. In this study, we have analyzed whole genome sequencing data for CRISPR-Cas9 edited lines from three independent experiments performed at three independent institutes. That is Yale University, Mayo Clinic, and Oklahoma Medical Research Foundation. For the experiment performed at Yale University, we got iPSC line derived from skin fibroblast of a male sample, and CRISPR editing experiment was done to introduce a loss of function mutation on FOXG1 gene. After that, three lines were picked. One of them were control line called B10 CTR, and two edited line called A4 and A8. To look for the unintended edits for these samples, we looked at the copy number profile and we saw there is an abnormal event on chromosome 20. So there is a deletion on control sample B10 CTR and edited sample A4, but the deletion was not present in the A8 sample. And to confirm this event, we looked at their heterozygous SNPs in that region, and it confirms the event. We also looked at the overall SNPs distribution for these three sample set, and their allele frequency distribution is close to 0.5. And then we were wondering that when the control line has the deletion, then why the A8 line has normal copy number for that region. So to investigate further, we did a whole genome shallow sequencing for the parental line, and then we found out that this deletion was present in the parental line in low frequency. So then it concludes. So we conclude that this event is not due to the editing experiment, but it's kind of it comes from the parental lines only. So the reason is that while picking up the cells for B10 and A4, we pick the cell that has the A4 that has the copy number deletion on chromosome 20, but in the case of A8, we didn't pick the cell that has the deletion. And we saw similar CNV events for both Mayo and Oklahoma experiments. So for the Mayo experiment, we got three edited line called CP58114, CP58836, CP60140, and one parental line called AA7. In AA7, we saw two low frequency event, one duplication on chromosome 1 and another deletion on chromosome 14. So this low frequency duplication on chromosome 1 become is prominent in CP58836 sample, but not present in CP58841 edited line as well as in CP60140. So similarly, that deletion on chromosome 14 is not present in CP58114 and CP58836, but it's present in a good frequency in the CP60140. And for these samples, we looked at the common SNPs, and we saw their allele frequency distribution is close to 5. And similarly, for the Oklahoma experiment, we got one edited line and one parental clone and two edited two clone from the parental line. And we saw two low frequency duplication, one on chromosome 1 and another one on chromosome 20. So the duplication on chromosome 1 is not present in any of the other clones, but the duplication on chromosome 20 is present on edited clone but not in the parental clone status. And we also looked at their allele frequency distribution, that is also close to 0.5. So so this kind of tells that this kind of events is kind of pretty common in CRISPR-Cas9 samples, and we should be aware of this kind of event while doing the experiment. So, to conclude, our study demonstrated that culture clones selected after the DNA editing experiments are not isogenic, and due to clonal selection and during editing experiment is a large and underappreciated source of genomic variability, and one should be aware of such genomic variation while doing the genome editing experiment. Thank you.